Our next speaker is Dr. Yuan. Kay is a lecturer in machine learning and computational biology at the School of Computing Science here at the University of Glasgow. His main research interest is developing novel machine learning and statistical methods for genomic and image data to answer questions in cancer and virus evolution. Dr. Yuan is presenting his work today on self-supervised learning for the discovery of novel morphological clusters linked to patient outcome and molecular phenotypes. Dr. Yuan, what is yours? How are you going to use this?
Okay. Thank you very much for the invitation, and very happy and excited to talk about where we're working on in the cancer domain using self-supervised learning. So to start, I wanted to give you a bit of a background of the story. Okay, so the cancer generally is an evolutionary process. It's a process that's similar to the virus evolution that we've observed in the past 2 years in SARS-2. It happens. It's basically a time course where the tumors start to accumulate in different types of mutations, and those diverse mutations allow certain population of cancer cells that you have already seen in previous images that may acquire different survival advantages to grow faster than others. So, but the evolution has another side, and that's the consequence of those genetic diversities. Here you're seeing the very early birth of the evolution theory. These are Darwin's ventures, where you see that clearly the beak of those birds are different from each other. That gives rise the idea of, okay, there's a phenotypic difference between the species, and the underlying cause of that is the genome is causing give rise to this diversity. But for tumors it's very the corresponding phenotypic diversity in tumor is not as well understood. I think the main challenge is that lack of systematic annotations for those wonderful H&E images that Chris has shown you. Although they've been hundreds of years, but actually the information content in those very large images is quite amazing. Here I'm just showing two small regions of the same lung tumor, and you see that there's clear difference between the two, but to what extent that these differences mean anything other than a clinical label that is imposed on the tumor. For example, a patient can survive or die within 5 years is very much unknown. So that present a tremendous opportunity, because there are a lot is unknown about the images. But now the development of deep learning gave us the new tools to learn new features from the images. So it's a lot of the exciting new research coming in this field. One example is that can you start using those routinely diagnostic image? Remember, those are the images that every single cancer patient is going to have, and use those to predict the genetic complexity of that patient. That information is not currently routinely available in the clinics. So the setup is really fairly standard. You have a training set where you know that for a given patient, its image and whether the genetic mutations that you have been sequenced to obtain. So this is all good that you can achieve some good performance. I want to present a particular challenge in the machine learning is going to face in this problem. So what you're seeing here is a measurement about the percentage of mutation detected in tumors across all cancer types, about how many of those mutations are deemed to be only exist in small part of the tumor, not all of the cancer cells. So you see that across all cancer types, and then ranging from 0 to very high percentage, right? You have lots of, basically the story that all of them has large number of mutations only exist in tiny fraction of the tumor. So what that means with the previous supervised learning task is that in the very unlikely scenario, you could be trying to predict mutation that is simply not exist in any cells that you see in the image. So that presents a very a sort of a standard challenge that you can put in the limitation of supervised learning. For example, these labels are expensive and rare, difficult to interpret predictions, and there's a lot of noise in the data. But in that extreme scenario, right, all you're trying to predict is noise in the image. Okay, so that's what basically sort of motivated our work to do unsupervised learning on those images to basically test to what extent we can extract useful information from those phenotype readouts. So this is a joint work led by a PhD student in my team, Alberto, and in collaboration with Aris Sirico's lab in New York. So what we set out to do is to, okay, can we try to solve some rather simple problem? Can we use a self-supervised learning framework that is recently emerging quite popular in this computer vision literature to answer a very simple question? If we present image being artificially preserved in different ways, can we train neural network to tell us they are the same thing? By doing so, network will give us a set of features that we use for us to better understand the diversity of the image. That is essentially what we're trying to do. And so we try that with a lung cancer data set, and we find is that in the representation we learn seems to be informative of different types of lung cancer. In this case, is lung adenocarcinoma, a squamous cell carcinoma. You don't need to know too much details about those, but they are actually phenotypic difference you can observe between those two cancer types. So then we basically trying to do is that, okay, can we find further diversity in those representation space that will allow us to know more. And then we did basically a state-of-the-art clustering method called Latent that's developed in single cell literature that's pretty good at dealing with high dimensionality of the representation. And we find out that there are certain structures in the space, and the clustering seems to be making sense. For example, you see in the network at the top, where these are more clustered telling one cancer type, and as moving down, you see different cancer types. Okay, so and then what we do is that you can actually represent the whole image in terms of cluster contributions, and then use this contribution to predict cancer type. And the result is that you can predict cancer type very well, and then it also competitive with sort of end-to-end deep learning approaches. And the benefit of this in terms of interpretation is that once you get those clusters, you can examine the contribution of a cluster with the prediction question in the traditional method there. We can in this case, you can assess odds ratios. And then another benefit with our clinical collaborators that you can pull out those clusters and then see what are those images in each one and check whether they make sense, right? And here are just some examples of the specific clusters to different cancer types. Okay, so now we set out to answer the question, okay, they seem to be clusters, they are able to predict things, are they actually real? So we focus our analysis on one cancer type where we have lots of site annotations, which can allow us to interrogate them in different ways. I'm just going to work you through some of the key analysis we've done. So again, we try focus on this adenocarcinoma cancer type. We try to find different phenotypic clusters. And then the first question we asked that can they predict survival? By the end of the day, those images are primarily used to predict prognosis. And we find out the same, they are competitive with existing approach in the performance of predicting survival. And we can interrogate which phenotype morphological cluster is contributing higher risk or lower risk. And then we ask a different question. So, okay, can we use the very same cluster defined in one data set and then do a sort of a zero shot transfer of in a different survival question in a different cohort. So we did that with an independent data set. This time the survival question is actually, can we predict the recurrence of the patient, recurrence of the tumor in a different cohort? It turns out that this very same cluster can actually do that pretty well. You can see in this nice UMAP prediction, the higher risk and lower risk group in terms of a recurrence are clearly separated in most of the cases. And then this analysis, due survival question and due cohort analysis allow us actually to identify something new in this domain, that is the local morphological patterns that are both contributing high risk of death and also high risk of recurrence. So this is something new. So we set out to further characterize actually, you see this general patterns, we can have a feel about, okay, there's some difference, there's some cluster here. Can we say that more quantitatively? So we employed a nucleus classifier. This is a state-of-the-art nucleus classifier we're showing on tons of cells. So what it does, it tells us for each cell in the image what type of cell it is. So we can make some statistical statement about whether a cluster is enriched or depleted in a certain cell type. And then we can look at all of our clusters and their relative relation in our latent space. On top, you see the UMAP projection of all our panels image, and see that within this latent space, if you just color code them in terms of enrichment or depletion of certain cell types, you can see that in the sort of left region, these are images enriched of tumor cells. But in moving on, the center region seems to be with lots of immune cell infiltrating those areas. At the same time, there's a large overlap between immune cell infiltration and lots of cell deaths. And that telling us these are the regions where you have active immune trying to fight off the cancer cells, and they are also certain degrees of success of that happening. So you can see that for those clusters, that's really what the heat map down there telling you. So those clusters that are sort of in uniformly reporting good outcome, and clusters are uniformly reporting poor outcome. For the good outcome clusters, you see enrichment in tumor cells, but also enrichment in the immune cells actively infiltrating, and also lots of dead cells in the image. That tells you why they're good for those patients. And then further, what's more interesting for our collaborators now, what else can those phenotypic similar regions tell us about the underlying molecular profile of the tumor? So what we did here is doing a large association with a lot of molecular signatures that you can extract by doing sequencing of the DNA or RNA or other thing you have. So what we see is that again, those clusters, the good outcome clusters and poor outcome clusters are associated with different type of molecular signature in a way that makes a lot of sense. For example, the poor outcome clusters are largely associated with signals that you may detect from DNA, for example, higher mutation rate, a lot of DNA damage can be detected there. And also, if you look at the good outcome clusters, you see the enrichment turns to be focusing on the immune composition of the tumor. That means that also basically in line with what we have seen previously, where those clusters are telling you that the immune population, the immune system is working very actively here. And then for each of those, you can interrogate, okay, the actual scatter plot supporting those correlations. And what's interesting here is that although the overall, in these two cases, although the overall correlations are low, but actually it is because those particular, this particular morphological cluster is less represented in a lot of those patients. But actually, when they have a significant representation, the correlation is much stronger. So this gives us a whole host of new hypotheses that can be further addressed using this the new knowledge that we've learned. So to quickly summarize, I'm just getting there. So you can extract a lot of useful information from histological images using deep learning, and the self-supervised learning approach that we have at least tried out is quite effective in doing that, and the clustering strategies need to be quite effective. So I wanted to do a advertisement here. So if you're interested in this type of work, that we are actually taking this further to do further using new technologies to do more detailed assays on those cluster we identify in a variety of different contexts, for example, pancreatic cancers, lung cancer, and also in the context of virus and host instructions in chicken. So with that, that's the, I'd like to thank you very much for your attention. That's the relevant references.
Thank you, Dr. Yuan. There was a lot to unpack there, and we have time for maybe one or two questions. Related clusters to Barlow twins. Maybe if no one else has a question right now, I want to start off with something that I have encountered while trying the same sort of problem. How would you make these clusters invariant to basic transformations, rotations, translation, that sort of thing? Because if you just vectorize a 2D image, I always encounter problems with that.
Thank you very much. So a lot of that has been already taken care of in the self-supervised learning training, because those two set of artificial augmentation we're doing involve those rotations and changing the color scheme, a whole bunch of other procedures. And they didn't talk a lot about those the details of self-supervised learning. And it seems like the Barlow twins objective, which is trying to push that your representations learning something that is invariant with those imposed augmentation, will minimize the part that is changing with respect to those augmentations. Seem to be playing a big positive role there.
Thank you. I might have to ask another question in the break because we're running a little over time. So thank you once again, Dr. Yuan.